Hi, my name's Angela Riveroll, and I am in Prince Edward Island, Canada, and I'm a research scientist at the University of Prince Edward Island. I'm a microbiologist and immunologist, and got interested in Python because we started working more with proteomic data. And I wanted to find a tool that would help me to efficiently um, analyze the large volumes of data that we've started to generate. So I think I'll start off with a sample of what our raw data output looks like. Um, in this case, we were looking at stem cells and we added various types of mediums to cause them to differentiate either into bone cells or adipocytes, fat cells. And we did each of these treatments in triplicate and wanted to look at the differences in protein expression profiles between those three conditions. What you see here is the mass spec output, a portion of it. It actually is 60,000 lines roughly, and here I'm only showing 2,424. I took a sample of the data. And what you see here is we have accession numbers that represent full length proteins. With mass spec data, this is generated through analyzing tons of peptides that are identified through the process. So I'm not really interested in all those lines. I'm really interested in line 2, 226, and none of the lines in between. So I needed to find a way to be able to select rows that contained only these accession numbers. And also, I'm really not interested in all of the columns all at once. I'm more interested in this case in the mean abundances for each of the particular conditions. So my control and then my two conditions, and these are the mean values. So I'll skip over to PyCharm now and run through how I managed to cut out some of this data using some of the um, different libraries. So I use pandas a lot, imported that as PD. Here's the file name that I wanted to look at. And then basically I just created the variable or the container to hold that data once it was read. I initially wanted to use CSV files, but each time I tried to convert this data to a CSV file, it would just crash Excel. So then I decided that I'd have to be able to read the data using Excel. And then, as I discussed, I just wanted to select certain columns of interest, like the accession number, and then some of the other columns. And then what I did was I just then used the to_excel method and exported it to a new file called select columns. So that's basically step one. And then step two, I wanted to then be able to iterate over this data that we got from Excel. And this was a little more difficult than I thought it would be, but luckily there's lots of communities online to help, and I realized that I could use this open um, PyXL to be able to iterate over my Excel sheet. So I use this particular library, and I use the method load workbook, and opened up the file that I created using the first script, um, defined columns, and as the worksheet that I wanted to work with. That was what I named in the previous script. And then I wanted to look at define what the maximum amount of rows and columns were, so that then I could iterate over those rows and those particular columns to then gain access to the cells. And this is really important. This is a challenging part for me to figure out how I could actually read the data, but this dot cell method the dot cell method open that up so that I could then use the regular expressions library to find my particular pattern. And, I just use this method where it's the expression would have to start with F and then have 1, 2, 3, 4, 5 characters so that I could pick up these accession numbers in the Excel spreadsheet, compile those patterns, and then I said if it matched, then I would append those particular matches to a list. So that was done, and then what I did next was to this is a part where I'd like to improve this program in the future. I used this method to identify my indices so that I could then use the indices in this iLoc method to cut out the particular rows. So if I hit data handling 2, yeah, you'll see in the output that I was able to first see that these are the number of lines I had. These were the number of rows, columns. I got my list of accession numbers from this particular file. And then I got the IDs or the indices, for the accession numbers that I could then input into the iLoc, and then I could then generate a new Excel spreadsheet now that would contain the columns, just as this, the columns that I wanted, as well as the rows. So you could see that I had now a 10 by 10 instead of a 2595 by 10. So then I then moved on from there to my next script. And in this case, I kind of made the assumption that if I subtracted my mean data for the cells that had converted into bone cells and the cells that had converted into fat cells from my control data, and I ranked that, then I might be able to tell where the major differences had happened. And so I just wrote a few scripts to be able to do that. So for example, key parts is data frame 2. I created a new column in my file called mean difference OM, and I said that that would be equal to subtracting the abundances from of the control, the abundances from the osteoblasts from those of the control, and then I created another column subtracting the data from the fat cells from the control. And the other thing is, these changes can either be an upregulation or a downregulation. So when we make the subtraction, I can either have a negative value or a positive value. I really wanted to look at just the magnitude so that I could rank the differences based on just the size of the differences. But I also wanted to um, not lose the information on whether it was upregulation or downregulation. So I use this Boolean condition to indicate whether the differences were positive or negative. So I said, give me another column, and if the mean difference that we got for either the osteoblasts or the fat cells were greater than 0, then I should have a Boolean that would say true, and then if it was not, I should end up with false. So I created these new columns, and for also looking at the absolute values. So I was able to use the apply function. So if I went apply and then this ABS function on this particular data, then I could end up with a column that would have the absolute values, and I could then rank them. So here's where I ranked them. I used sort underscore values method, and I said I wanted it to be sorted by the mean differences for OM, and I wanted it to be descending. I wanted it to start from the highest and go to the lowest, so then I just set ascending as false. And so then I created two more Excel sheets, one with rank column rows for OM and one rank column rows for AD. So I guess I can just skip out to show you what that looks like a little bit. So if I go to ranked, then you'll see that what I ended up was a spreadsheet was generated and with that name. And you could see that I ended up with the absolute values. And you can see one item in here was false, and that's because it was a drop in protein production and not an increase. And I'm thinking this will help me in the future when I'm looking at data visualization to be able to maybe color code them differently if it's upregulation or downregulation. So now that I had my ranked values, so I'll go to this particular Excel sheet where I kind of gathered my summaries. Now I kind of had my ranked values and I had my mean difference, but now I wanted to actually run some statistical analysis because I wanted to be sure that these differences that I saw were actually true differences and not just errors in the data. In order to do this, I could no longer work with the mean data that I had gotten in the first file, so I had to request some more data and to get individual values for the individual conditions. And so that brings us to this spreadsheet where you can see that instead of having just the three group columns, now I have for the individual. So S01, S02, S03 would have been my controls, and then 9, 8, and 7 would have been the fat cells, and then 6, 5, 4 would have been the osteoblasts. So the next phase of the work is actually working with this particular new file now and wanting to run one-way analysis of variance on this particular data. So that brings us to the other data handling software. So then I could kind of recycle some of the code I'd originally written. Same thing using pandas, opening the file name, reading it, picking out the columns I cared about. Again, getting the IDs and pulling them out. Interesting is that the data that I got from the second pull from our contractor was not as stringent as, or was more stringent than the other poll. So not all of my accession numbers were pulled out. So that's really interesting. I'm going to have to follow up on that a bit. But data handling 4. So perhaps I removed my printing. So I'm going to remove, this hashtag so that we can see an output. And I'll give that another run. Handling 4. Oh yeah, this one takes some time because I'm actually running through all of the lines of this data, which you can see is now a lot larger. So it's more like 54,000 lines of data. So it sometimes gets hung up here a little bit. And this is another reason why I decided to create multiple scripts instead of just one, because some would take quite a bit longer to run. Okay. Okay, so this script is taking quite a while to run. Oh, there we go. Okay, so this is the older one. Okay, so you can see there it pulled out the 5 rows that I wanted from that particular large file. So then we can move on. So now that I had this particular data, then I could run my ANOVA. So I had to transform my data because the way the ANOVA works, it requires that the control and the various groups be put as rows instead of as columns. So data handling 6, all right, actually data handling 5 was a very simple script to transform the data. So much of this is the same. I renamed some of the columns to make things simpler into just converting these big long labels to just SM. And then I basically use this df2 dot transposed. And if I print this, run data handling 5, I end up with this is the initial data with the rows on top, and then they ended up being on the other axis. Now, I did have quite a bit of trouble with this one, and I think I have to do some more research in terms of indices. But in order for me to be able to run a box plot and ANOVA, I have to get rid of all these indices here. And for example, this was generated as a name and these as objects, so it made things a little bit complicated to get the table in the format that could be recognized by the next scripts. So I ended up exporting it to Excel, and then I did have to make a minor modification, that is to remove these indices in Excel. So So here's my version 2, just to give you an idea of what that looked like. So you can see, in order for me to be able to run a box plot, this took me a while to figure out. I had to have just these as the columns, and then I created this new column called groups because I wanted to then group the data by these particular tags. So that brings us to the final script. And here is where I opened that particular file I just showed you, just as I did before. And then I used other libraries this time, including Matplotlib. It's grayed out now because I have the actual showing feature shut off for now, but I think I will remove one of them to just show one of the plots. And then I used stats model to then run the ANOVA, and of course pandas to read all the files. So if I so basically I had my data frame and I used the box plot function, and if I hit run, it sometimes gets hung up on the box plot, so this is why I use the hashtag to just silence it momentarily. And you can see that for one particular protein, that's the accession number, you could see if this was the control, this was the mean, this was the mean for the cells, mean protein level for the cells that were differentiated into bone cells, OM, and then these were the ones that were differentiated into fat cells. And so, you know, this helped me to look a bit at the distribution of the points and the errors. But as a next step, I need to actually run the ANOVAs because I want to be able to determine whether the differences between these groups are actually significant, whether the differences between the replicates is, you know, as large as the difference actually between the groups. So to run the one-way ANOVA, I then going to silence this plot because it seems to hang everything else up. I'm going to just run the one-way ANOVA. And you can see that I end up with, these are my groups, so these are the various indices, and you can see that I ended up with the p-values to suggest whether or not the difference was significant. To run the one-way ANOVA, I used the stats module methods, and I said that I wanted to, you know, look at them by groups. My data was the data frame that I created, and I wanted to use the fit math function to fit this data into a particular ANOVA model, which came from SM stats. library up here. Um, so then basically then I just requested a print of the ANOVA tables, and that's what I ended up with. So in summary, I was able to rank the different proteins or and their accession numbers based on the largest mean differences. I was able to indicate whether it was upregulated or downregulated in this case, and then I was able to run the ANOVA to look at whether these differences were actually significant. There are a few areas definitely where I'd like to improve the automation on this. In particular, there were a couple places where I had to actually where I had to actually type in the accession numbers. I'd like to be able to just find a way to get that a better way to get the indices instead of this method. And I think if I can then grab those indices and get it to populate a list, and I could just iterate over that. So these are just some areas where I just didn't know the pandas library well enough and kind of got stuck and found this way out, even though it might not be the best way out. Um, and the other thing that will be interesting is at the moment I'm using boxplot to kind of look at the data, but in the future I'd like to, when I end up with thousands of these accession numbers where I see that I have a significant difference, I'd like to then start grouping these by systems biology function, and then be able to display in some way whole systems being upregulated or downregulated in response to a cell determining its cell fate. So that's all for now, and I definitely felt I learned a lot during this course, but still realized that there is a whole lot more to learn, but it is definitely a powerful tool to try to master. Anyhow, thanks Professor Parker and all the TAs. It was a lot of great learning, a lot of sleepless nights, but a lot of great learning. Thank you.